Thank you, Sarah. As you all know, we're going to be recording this. I believe we have captions enabled at the bottom. So if English is not your first language, you are welcome to use the captions to help you follow along with us. We're so happy to have you all here. Welcome and thank you all for coming to our first installment of 2023 of the Amgen Biotech Experience Teacher Roundtable Series. I'm Jessica Juliuson. I'm the Director of Community and Strategy for the ABE Program Office. And as always, it's an absolute pleasure for me to be able to host this roundtable today. For those of you who are new to this series, these roundtables are designed for our incredible ABE teacher community from around the world. And it's a chance for them to hear and learn from experts and from each other, from other teachers, about topics of specific interest to science and biotech teachers. So we hope you find this series valuable. Please feel free to tag us in social media if you want to share your thoughts. You can tag us with @ABE Prog Office, and I think thank you, Sarah, for pasting that in the chat so you can find it there. We also invite you all to say hello, introduce yourselves, and say where you're from in the chat as we proceed. This roundtable is going to be recorded and posted on our website, and if you have registered, you will also receive a transcript of the discussion and a copy of any materials that we share today. We will have some time at the end of this hour for audience questions. And so at any time, if you have a question, you can save it for the end, or you can just type it in the chat and we'll keep our eyes out and make sure we're collecting all of your questions as we go. And I do want to begin by first sharing the news that unfortunately one of our roundtable panelists, Deb O'Reilly of ABE Massachusetts, did have an unexpected emergency come up and she's unable to join us today. She has been a wonderful leader in ABE and was recognized as Massachusetts Outstanding Teacher for 2021 by the National Association of Biology Teachers here in the US. We will miss her today, but we are very fortunate to have another outstanding teacher from ABE Massachusetts, Dr. David Welty, who is joining us in her place. So a huge thanks to Dave. So I'll jump right in by introducing our panelists. Mary Lou is a high school teacher at a public high school in Massachusetts, where she teaches honors biology, AP biology, and biotechnology. She's been teaching for over 12 years and is currently a teacher in residence at LabXchange. Mary is interested in integrating opportunities to build science skills in person and with digital supports at all levels of science. And I personally would like to add that Mary has been an incredibly valuable contributor to the ABE program overall, assisting her program site in Massachusetts, as well as the International Program Office with piloting and development, feedback, and leadership at so many levels. So we're really excited to have her back with us today. And our wonderful stand-in today, Dr. David Welty, has taught biology, chemistry, and physics over his past 21 years of teaching in Fairhaven, Massachusetts. Recently, he has taught introductory pre-AP biology, college prep chemistry, and AP chemistry. And he was just telling us that he's also teaching organic chemistry for the first time. So not a small amount on your plate, Dave. Dave has a PhD in molecular biology and biochemistry based on protein expression, purification, and regulation of proviruses in the bacterial genome. He's been an ABE teacher for over 10 years and collaborating and co-creating with LabXchange since it began. David, a huge thank you for joining us today at such short notice, and we're so pleased to have both of you with us today. So we'll begin by getting to know a bit more about our panelists and their journeys as educators. So Mary, for those participants who don't know you, please tell us a little bit more about your pathway into science teaching and the role you're now playing with ABE Massachusetts and with LabXchange.
Sure. I'm originally from Ohio, and I had wonderful science teachers and became very interested in science. When I came to college, I had wonderful professors there as well who kindled that interest in science, but I also dabbled a little bit in teaching. In the public school, there was like a co-op program with the Graduate School of Education. I could earn my teaching degree as well as my undergraduate degree in science. So I did both the pre-med and the education at the same time. I found out I really loved the teaching aspect of it, and I've been there ever since. So I've been in the classroom. I went back for my master's in education, but I've been in the classroom ever since that. And then in terms of ABE and LabXchange, I connected to both of them when I was in college. There was a lecture series for science teachers hosted at the university, and I hooked up with other teachers there. and discovered the ABE program, and then brought that into my classes. And then when LabXchange started, it actually supported this ABE experience, and I got connected with them that way. So I'll
Thank you.
pass it
And
back
so
to you.
Mary's been such a great advocate for the teacher voice in how LabXchange has developed assets and saying this is what teachers really need and use in the classroom. And so we're excited to be working with Mary, and I know LabXchange is as well. So Dave, how about you? What was your journey to education, and how did you get involved in science teaching and then eventually LabXchange?
Well, when I was, well, one of the interesting things is Mary and I grew up in Ohio, about 30 miles away from each other. So, and we didn't meet each other till we're out here. So both two Ohio transplants. But when I was at Ohio State, I went into the career office and said, I want an internship. And so they put me at the National Cancer Institute at NIH. And I just enjoyed the whole process. And so I was a technician there for 2 years. This was before PCR came out. And when I actually joined the lab after graduating, my job was to create PCR for the lab, to convert Southern blot techniques over. And eventually I realized that if you needed to do anything in science, you needed to get a doctorate. And so I ended up at Georgetown University, and with molecular biology as my foundation, but I really got fascinated by protein chemistry. How do the side chains interact and all of that. So I eventually dabbled into becoming a protein chemist. While I was there, I was teaching medical students, and I had two interesting students. One had complete recall of everything, but couldn't sort of think things through. And the other one was really good at thinking things through, but couldn't memorize anything. So not knowing anything about education, I was developing two different lesson plans, so to speak, for two different learning styles. And that just stuck. And eventually I worked in a pharmaceutical company for a while, but I had a daughter. It was tough balancing a family and working in a lab. And I basically made the decision to save my family, to go into education. It seemed to be more friendly. I could actually be home. Little did I realize that I would be spending about the same amount of hours in education as I was in research. It's just that I get to bring it home. And so I get to sit on one end of the couch and my wife is on the other end, and I'm grading, making lesson plans, but we're together. Whereas before I was in the lab. And through PCR, I had a couple buddies in the area, and they said, hey, there's this Amgen group that's kind of giving out equipment and everything. I'm like, well, tell me more. And so he and I and a couple others, we got involved, and eventually just said, okay, I'm all in. And so I became an ambassador. And I work with Sven Henrik. He is a Harvard professor at the time. He did the labs for the undergrad. And he and I kind of work cooperatively, where he comes up with the ideas and I kind of help him bring it down to hit the high school level. And I have the spectrum of sort of beginning learners, novice learners in biology, all the way up to organic chem. And it's been a nice relationship. And one of the things is, you know, you may have heard, is I'm deep into chemistry. So my motivation is to help LabXchange get more chemistry into their biotech. I mean, it's pretty biology friendly. They're becoming stronger with chemistry, but my passion is really to help them get more chemistry in. And so that's where I am.
David, such an interesting story, because hearing about how you learned to teach with all of these different audiences, you know, starting with adults and then kind of with this range of learners, it's just, it's interesting because it sounds like you kind of started off thinking about differentiation, which is something we'll ask you about a little bit later as we keep going in the roundtable. So thank you both for being here. I love the stories always of how people ended up in the classroom. And so I want to ask you now, online and hybrid learning was obviously a fairly hot topic even before the global pandemic, but then once COVID hit, and classrooms really ground to a halt, particularly science classrooms that were so reliant on hands-on experiences for their students. How did you see LabXchange supporting you through that experience? In what ways did you see it as a game changer? Do you see it as a way that it can alter how teachers think about biotech instruction? And let me start with you with that question, Dave, since you were just talking.
For me, it's biotech and chemistry is not something the student can visualize. And so my whole task, my challenge is to allow and help students find ways where they can see what they can't see. So I've used a lot of the assets, the ones that are sort of simulations of not only the molecular world, but trends to help students visualize that. Through the pandemic, when I was using more LabXchange stuff remotely, I realized that they still need that guide on the side. And so I use a lot of scaffolding, and I pulled it more into the classroom. I have found that for my students, they need to be able to have the digital, but they also need to have that hands-on group interaction. So I'm really using a hybrid in the sense that they're using the digital, but they're working together to get that social. I don't know about you, but I found that my students really took a step back and being able to work together. And when we first, in my school, I'm not sure how it was in your schools, but we didn't have any hands-on labs. We had no group work for 2 years. And so we had to reintroduce students to a lab setting, to working. And so I've kind of taken the power of LabXchange's assets and simulations and then scaffold it with my background so that the students can work together to come to conclusions and understanding.
That's really interesting. And I've heard a lot of teachers talk about that kind of the incredible value of having those scaffolds, and you can use them at different points in time. And I'd love to hear a little more about, how you use it to support collaboration. And so is that something you specifically use LabXchange for, or just is that something you're teaching in person and then the LabXchange helps?
Well, the LabXchange, I primarily use the assets, and then I'll set up either a Google form or a Google sheet. A lot of times I collect data. I find that my students are deficient in spreadsheet use. So I use sets that produce data that they can actually capture that data and then produce graphs and statistical analysis. So we do a lot of that. Primarily where I use it through the lab format, where we're learning like protein purification of the column. That's more independent work where I'll introduce that so they can get a sense of what the lab is going to be and the skills, and that sets them up so that they can then work cooperatively at the lab bench.
Great. Thank you, Dave. And for Mary, how about for you? Did it change the way that you found you were teaching science when you started using LabXchange? How did it support you during the pandemic or before or after?
To me, the biggest value that LabXchange added, in addition to the visual components, have good visual supports everybody, right? It's just a good model for helping teach concepts. But to me, it was the flexibility. No matter the year, no matter if I'm teaching, like every year, somewhat flexible to the students who are in the room, to the levels that they bring, and particularly like pre and post pandemic. Like now, students are in all kinds of different places. I felt like students, didn't all reach the same levels when they were online learning solely during coronavirus pandemic. So now I think it's important to help support them by like diversifying the options available to them. Like having that sense of choice not only better engages them, I think, but also helps support them with their needs, whether they have an individualized learning plan or not. Because you can customize and move things around, I can add in things that I know, like that I'm going to use and keep it sort of as a record for students who might be absent or miss periods of learning. but also just have a record for anyone who wants to go back to it later. So I found that flexibility and customization part of LabXchange really helpful.
And I know some of what you're describing there is based on that use of the classroom feature and the classroom function of LabXchange, which I know you're going to show us more about later on when you're doing show and tell. But some of the things I'm hearing from both of you are that it can change science instruction by providing greater scaffolds and by providing some of these visuals and ways to see things that you don't see. I love that phrase, Dave. And also as a way to kind of provide different experiences for different learners. And so that kind of brings me to my next question, which is about equity. And as you know, equity and access is a high priority for the Amgen Foundation and for the ABE Program Office. And the LabXchange team has been very involved in promoting equity and access in STEM. And so, Mary, I know you have a pretty good sense of this being so closely involved with the team. What kinds of assets and resources does LabXchange offer to teachers that might support greater equity and access in their own classrooms?
One component is just the availability of the assets in different languages. So one big push on LabXchange has been to translate the ABE content and other content into a variety of languages. I think right now it's available in 14 different languages to provide support for students everywhere. In terms of like some of the background details, they're dedicated to making it free so that everyone has that level of access, and mobile first for quick loading times so that they can make sure that everyone can use it regardless of the kind of technology they bring to the table. But to me, another component that leads to this equity in terms of science learning and thinking is their simulations. So their simulations are really science skill focused. So students can make mistakes, they can go through and change different variables. So instead of just having like a passive experience, maybe they're absent or they don't have access to lab materials, they can still get some of those science thinking and science skills virtually, putting them on a better playing field for whenever they want to access more science.
That's so important, and especially as we think about digital equity. I mean, that's obviously something that plays into this as well. And I invite anyone who's in our audience, if you have had experiences with using LabXchange to support greater access, greater equity for the populations that you work with, please share them with us in the chat. We would love to hear more about them. And Dave, how about for you? How have you used LabXchange in a way that provides greater access or diverse access for your students?
Well, one of the main reasons is representation. If our students can't see themselves in the field, then they don't see themselves in the field. I mean, to speak. And I was working in a inner city summer camp, and we were trying to help students who are ESL, English second language speakers, see themselves as scientists. And I was able to go in and pull out narratives of scientists around the entire world, scientists of color, different pathways in. And we did an interesting experiment. And I was working with a scientist of color, and she and I were standing together in the front of the room, and we asked the question, which one of us is the scientist? And we were hoping they'd say both, but having a white beard, white hair, being male, they all pointed to me, and they didn't see the other scientist as being one because being a female of color. And so that was really striking, that we need to do a better job of letting kids know that scientist is not the white male, it's everybody. And so that was just a really powerful, and I don't know if you've noticed it or not, but ABE's diversifying their characters in their simulations. So it's just not a white male there. It's female, different hair. I mean, very subtle, but it's there. So they're trying to change their entire representation. And again, the narratives that you can go in and see the different postdocs and everything are also extremely powerful. I remember some of the students saying, wow, I didn't realize there were scientists that looked like me. And it just sort of changed my entire perspective. So the narratives can be extremely powerful for us to change. So
Thank you for sharing that, Dave. And I appreciate too the fact that this is a work in progress for all of our organizations in terms of transforming the messages that we're sending to students about who they are, what they're capable of, the fact that they can do science, that they can step into some of these pathways into science, and that LabXchange has been part of that effort. And so it's great to have so many more resources than used to be available at your fingertips as a classroom teacher for those things. I appreciate both of your answers to that. And I see that we are at time, and I don't want to step too much on the time for show and tell, because I know many people are anxious to see, what does this look like? What do you actually use with your students in the classroom? And so for the next, half hour or so, we're going to be asking our panelists to share their screens and walk us through some examples of how they use LabXchange with their students. I would love to invite all of you in the audience to share questions in the chat. And if you have ways that you use LabXchange that you think are particularly exciting or engaging for students, please put those in the chat as well. And we may ask you to speak to one or more of those if we have time at the end of the roundtable. So please share with us in the chat. And Dave, at this moment, I'll mute myself and turn the screen over to you.
Okay. There we go. Can everybody hear me?
Yes.
Okay. Well, unfortunately, I'm on my wrong screen. So give me 1 second here. Thought we'd had that all worked out. Nope, I had it there. There we go. Okay. Okay, so this is an example of how I adapted a presentation that I was doing for students. And what I'm doing is I'm using multiple teaching techniques within this presentation. And a lot of the images that I pull, I've gotten from LabXchange. So one of the things that I want to make sure that my students know is modeling, and what is our best idea of what the molecular world looks like. And so here is a typical
Hey,
model.
Dave,
Yeah.
I see cell membrane transport. Is that what you're sharing?
Yeah.
Oh, okay, great. Just checking. Someone mentioned in the chat.
Okay. Do you see this diffusion?
No, it says cell membrane transport.
Okay, so I'm on the wrong screen. I hit something I wasn't supposed to hit.
If you stop sharing, Dave, if you stop sharing and then start again with we were joking before the roundtable that we're all rusty on these things now, but
Okay,
we'll get
do we
there.
have to, is diffusion up right
Yes,
now?
now it's diffusion. Now it's
Thank you,
diffusion.
Mary. Okay, now how are we doing? Everyone can see the animation model? Okay,
Yes.
so this is an image that came from LabXchange. And this just gives students a better idea of what a molecular model would look like for a cell membrane. And whoa, I'm sorry, I'm having. Okay, and then so as the students are working, we're a one-to-one school, and as the students are working, they can sort of put their definitions in as we move along, and then we have embedded simulations. Okay, so here's a prediction: which way will the oxygen move? Again, this is a simulation that I pulled out of LabXchange, and so they can sort of make their observations and place them there. Again, another one. So this is a technique that I use a lot. It's called a See, Think, Wonder, and allows students to do a deep dive. And same thing, I'm having them mention 3 things that they see, 2 things they think, 1 thing that they wonder, and then what's the summary. And so when I hit the hyperlink, come on. There we go. Okay, so now you've seen we've gone exactly to LabXchange, and I can start the simulation up. Okay. And will work. There we go. And so the students are making observations. They can start to then see a phenomena that if I was just to do this in the front of the bench with red dye and some water, they're not going to be able to really get the sense of what's going on molecular. So this is an example of how I took assets from LabXchange and embedded them into a presentation. Another way is that we can do pathways, okay? And so this is a Beers-Lambert and standard curve pathway that I created, again, using assets within LabXchange. And I can pick and choose things that I want my students to read, to participate in, to follow. And this would be for an AP Chem. And so there's a text. Here's an interactive. And it basically is going to take a student through the whole process of how a Beer's law would work. Okay. Let's see if I can move that over. There we go. Okay. And we can just asking simple questions, and they can move this with their cursor. Okay, how to read a standard curve. And so this basically gives them the steps that they're going to go through to figure out how to set up a standard curve and how to read it. And then there's some more images, and then there's this curve fitting simulation from PhET. So that's one of the things that I really like about LabXchange is they actually have many of the PhET assets, not only current, but some that are past. And sometimes I can't find them anywhere but on LabXchange. And so again, how to do a curve fitting. And the one that I use the most is the Beer's law, and I have not found anything that's quite as good as the Beer's law. And what's really nice about it is that you can adjust all of the variables in spectrophotometry. So we turn on the light and we start to read the transmittance. We can actually go in and I can apply the negative log to this transmittance, and I can get absorbance. And so it's just extremely powerful. I can change the path length, and this is just, in my opinion, really strong. Change the chemical substance. And so that's there. And then what my students do is ultimately I put one of my documents on that I've created, and I can put it right there on LabXchange. And so then they have the document there. Okay. So I was able to do this lab during the pandemic when my kids were at home. They could do the whole thing and they could actually do the lab. Now they would have to fill this out with another one, but it's right there. Another one that I was involved in the making of this one is Le Chatelier's principle. And so this is dealing with equilibriums. And what's nice about this is I have both an older and a newer version of it. We'll wait for this to load. And so as we run through, we can see the reactants changing in concentration, and we can see them reaching equilibrium. And so then it reaches a constant equilibrium. And so this is another one where I have students collect data, and they will then calculate what is the QR of the reaction. Is it still moving towards products, or is it going backwards? And so I use that, and that's one that was I was involved in, and we strictly needed something for Le Chatelier, so I had a lot of say in that. Again, I've buried some, not buried, but I've connected some Google Docs that I created that allows the students to sort of investigate what's going on with concentrations of the reactants in the products. And so again, that document's right there. So everything is self-contained, and I can create it just the way that I want it. And so that's where the tailoring comes in, the crafting, the scaffolding. And then I teach both organic chem and AP chem, and I've signed up for the end of the year to have the protein column chromatography. Oops, sorry, my lights just went out. There we go. And so this is one that they're going to use to learn about column chromatography. And so I use column chromatography to teach chemical interaction. What's kind of nice is Mary and I actually, way back in the day, made a video on how column chromatography works. So that's always kind of fun to show the students that, hey, I'm on the internet. And so that sort of brings me a little street cred there. And then we got the simulation for how protein chromatography works and how the amino acid side chains will interact with a hydrophilic or a hydrophobic. And so those are the main ways that I use LabXchange. Is I build pathways that then I share with the students, and they have sort of a scaffolded set of steps that allows them to learn a new technique, master a new technique, and prepare for a lab. So, any questions?
Dave, thank you so much for sharing that. And I know we do have a question in the chat that Mary, maybe you can address when you're going through your show and tell, which is about when you're actually creating the pathways like Dave was showing us, and you want to include an interactive from another website, whether that's possible to add or whether you can only use existing assets. So Mary, I know that you can probably address that when you're doing your presentation. So I'll turn the camera over to you and let you share what you have for us today.
Okie doke. I'm gonna just leave my video off because I know I've been a little laggy. So just in case if this helps with the smoothness, I'll leave it off, but I will keep an eye on the chat just in case. So I just went to labexchange.org. You can see the whole library here. I'm gonna share what I use for both my biotech and AP classes or honors classes with the ABE cluster. So in case you're not familiar, the library is basically just like a repository of all kinds of things. There are interactive components like simulations or scrollables like Dave showed off. There's text and video assets, and you can sort them and search for them either by the type of interactivity, maybe a subject area, a particular source that you're interested if you like some of those like FET, like Dave mentioned, or particular places, or just with keywords. So if you wanted to find, you know, the biotech cluster associated with ABE, if you search biotech, it'll come up. It is this cluster, and what a cluster is just what a cluster is just a group of pathways together, sort of themed, so that it can be shown together. And you can see there's a bunch of different pathways here. And each pathway is a series of learning experiences. So for example, this tools and technique one, this is for transformation, and it's completely editable. So I clicked the 3 dots that say clone, and it makes my own personalized copy. So this is what I do for my classes. You have 2 options. You could just use the URL link, share it out with your class. Anybody can access it. You don't even need a login. It's public in the public library, and you can use it as is. If you wanted to create a customized version, if you make a clone, it looks like this. So you can change what the description is if you want to personalize those learning goals. You can change, you know, the order of these assets. If it's a video, like this long video is 7 minutes, if you want to clip it, you can use the little scissors and show just a section of it. Or as someone asked, if you want to add things, so you could click this add content, and you could add your own content. You could add content from other places. If it doesn't exist in the library, itself, you can choose this add new content, and you can add in a link to something that you're interested in by just adding a document. You might add, a customized question if you want some formative assessment, or an image, something that you use already, a text. If I was linking to a simulation, it would probably be embedded either in a document or a text, is my guess. And students can access it as part of your pathway. So anything you add just kind of appears in your little list here, and students will see it as part of the whole package. Any of these customized versions end up as private to you because you're adding things that are yours, but you can still share them with your students. And you have two options for that. You can just share the URL at the top. That'll take them directly to this pathway. And that's one way to share it with your learning management system. Or you can also share through a class, and I'll show you how to do that in just a second. But I just want to sort of show off some of the features of things that I've added. So for example, like here, like a graphic organizer for students. I find that a lot of times, because the ABE sequence is over several days, they need a reminder at the beginning of the day to see sort of where they are in the pathway. So we start off with that graphic organizer. And then for both of my classes, the simulation is really helpful because in the end, if you're not familiar with these simulations, what they have you do is sort of walk through the experiment, but they also include opportunities to predict and reflect on your results. And they present the information at 3 different levels of detail. So if you are just starting this or aren't familiar with this technique, level 1 is a good fit because it has a lot more detail and description in the protocol section. If you maybe have a more advanced group, you can choose level 3, or you can let students self-select. It gives them some background information, just like you would before you start, about the technique itself. It gives them a list of all the things you're using. You can click on these for more information if they're not familiar with these tools. And then you can predict. So I thought this was great because it really gets at the heart of what this technique is by visualizing what's happening in the tube, right? Sometimes it's hard for students to picture, okay, I know I'm dealing with bacteria somewhere in there, it just looks cloudy. But this really drives home the point of what transformation is. I will go back. Thanks, Teresa. I see your questions in the chat. I will go back and show you again how to make a clone of a pathway. And you can't create a cluster, but when I show you the classes feature, maybe that'll help address what you're looking for. But again, the visualization is great, and you can make predictions. So students can select one. And then in the reflection, when they get to the end, they're represented with those predictions and can reflect back on them. So here's the lab scene. They can move all of these pieces. They can carry out, add things. There is a protocol on the side that tells them sort of a list of steps that they can do to complete the activity, but they don't have to follow it. I know my students kind of just explore and see what they can play with. And this isn't just a click-through experience. So when you get to results, it actually does carry through whatever you did. So if I didn't do anything in the lab, I'm not going to get any results, which is great. Because then I can have my students reflect back, okay, well, what could lead to this? Why didn't you get new results? Which steps might have you done differently? And it does show them the ideal results. So you can think about that as well. And then in the reflection section, it presents them with some questions, having them think through more about the technique. Now, let me just jump back to, here's our cluster again. And if you're from one of the ABE sites and want to connect it to the 6 labs, this tour section, if you click one of these, it shows you which pathways are associated with each of these labs and gives you a suggested order. But of course, you could use all of them if you'd like. It also shows you based on theme. So if you're just like, how do I look at the ones that have techniques? It highlights those ones. And then when you're wanting to clone, right, let's say I click on one of these. When it opens, you'll see there's some options here at the top to favorite. So that just will put it in a section on your dashboard so you can come back to it. You can assign it to a class, and I'll show you that just now. You can share it to Google Classroom or copy the link or share it socially. And then it's these 3 dots is where you find the clone button. You can clone any pathway that exists in the public library, or you could create a pathway from scratch, but cloning will copy this one. So just hitting clone creates a copy of it for you in your dashboard. It copies everything there and gives you those options to move things around, because you can see right now they're static. There are no arrows and no edit buttons, but the cloning feature, when it makes a clone, then I have all those options available to me to edit the order and sequence of things. And one thing I can't edit is the content of someone else's asset. So for example, this is by Welcome Genome Campus. I can't go in and like change the words that they used because it's their asset, but I can add context around it. So this notes feature lets me add a personalized note. So if I want to tell my students, okay, this is optional background, or if I want to give them a definition, kind of thing, I can add those things and they sit on top of the asset to help support students when they're experiencing the pathway, but it doesn't change anything about the asset behind it. So yeah, you can make all those changes, and that's one way I also customize class to class. So let me show you the classes feature. So you'll find the classes here if you go to dashboard. This is where all kinds of things sit. One is any content you've personally created. So when you create a clone, your cloned pathways will live here. You can add your own content if you want somebody to experience just one of these pieces, like a document or a text or a teaching guide. You could create a pathway from scratch. Upload some audio files like podcasts, or add assessment pieces. There's also favorites, so anything you star ends up here, so it's easy to find. And then the classes option. So this is great for a teaching context or for professional development. I've seen, we've run like webinars and professional classes this way if you want everyone to have access to resources. But if you click this create a new class, you'll see it will appear here. If you click on this, you can kind of see what I'm showing off to my students. And in my class, basically I can show off what I want to show them in the public view that I've posted. And then I can have things kind of waiting in the wings. Depending on when or the type of class I'm interested in sharing content with, I'll hide or remove these things sort of as I need them, and show only a few things at a time. It kind of depends. So you can't create a cluster that's a list of pathways, but you could basically do that in this class context. You could list a bunch of pathways that you're interested in sharing with your students, and they can access it through this class. To access the class, they just need to enter this code. So you can see this class code up in the corner is what they would add in when they go to their dashboard. It gives them the option to join. You can see anybody who's joined here, and you have access to let them in. So it is a private class so that you don't have to worry who's in your class. You can make sure that you approve them before they get in. And then it shows how your students interact with the content that you post. So you can see like different pieces here, shows how much they've completed, if they've completed it, how much they got correct, if there's an assessment built in. So you can kind of check in on students as they're going, and you can download that report if you'd like. I use it more of like a formative assessment. If everyone's working on something, I can kind of check in with students as they progress. And with those different learners, you can go through and assign them things individually. So here you can assign a particular piece of content. So if I wanted to differentiate for some of my students, maybe they've missed something in class that I want to assign them to catch up, you can assign them just a single piece. And then they get messaged it directly. You can check in on an individual student's progress. So if you only want to see one person at a time, you can see which pieces they've started or not started and share that back with them. Or you can message them directly if you want to chat that way. It sends them a private message. So those are some of the features of classes, and there is a discussion board on that as well, so that you can use that as well in a hybrid way. One thing I found is helpful, even if you're not using the classes feature, right? One more place to go. If you post your content here and just share the URL, it'll automatically track all these things for you. So students don't have to go to LabXchange and then find the class and then find this piece. If you just post this URL here, if you just copy paste the address bar URL, put it wherever you are putting anything for students like Google Classroom, then they'll have access to things in your classroom and it'll track all that progress for you. So this sort of customizes the URL too, so it sends them to this particular place. All right, I'll pause a second and see if there are any questions that appear.
I haven't seen any new questions, Mary, but I do have, there was a question that Teresa asked earlier that I think is probably worth talking about, which is when you're making a pathway and you want to include an asset, something like an interactive that from another website, can you do that?
Mm-hmm. How
do you add assets from other places?
Right. So it depends on the kind of asset that it is. Let me just go to my dashboard. If you are using something that has like an embed code, maybe you can add that in if you know some HTML tricks. But the easiest way to do it, and what I would do for my classes, would be just to add it as a text asset and put the URL where I want to send them, or whatever other instructions I want them to preview while they're looking at that simulation. So if I just click add content, if I find a simulation link here, and instructions, right, I would make it one big sort of text asset. And then when you go to save it, it will appear, whoops, let me, instructions. It will appear in your library, and then you can embed that into a pathway or share it out directly. So you can embed things from anywhere. They're just private to you. You won't find them in like the search bar, but you can find them in your dashboard.
Thank you, Mary. This is great.
Sure. Yeah, let me just share out also this remote learning cluster that they've made to help explain how to use LabXchange. If you're not very familiar with it, you can see it reviews all these things I've been talking about, how to create your online class, how to teach lab skills and using those simulations. There's also experimental design simulations that look a little different. It's more about dragging different pieces together to create an overall experiment. Like if you do a restriction digest, what's the next step? Oh, you need to run a gel kind of thing. So it showcases that kind of experimental thinking as well. And then also I wanted to just show, just in case you didn't see in the dashboard, whoops, let me content type cluster. I just wanted to showcase the different language components. So you can search in this filters on the side by different languages if you're interested in finding something that way. You can also, if you find something that you like in English and want to change it for your students, you can just click change and then you can highlight all the different languages. It'll automatically change the content pieces to the language that you're selecting. And then students can experience it that way. Yeah. And then in terms of if you're looking for extra things to sort of support this, these are particular pathways or learning experiences, but in the library you can search. So if you're like, okay, I really am interested in, you know, gel electrophoresis, but I'm looking for a way to assess my students. So I'm going to use content type questions to see what's in there for gel electrophoresis and search that way. You can kind of filter by sort of your interest if you're looking for interactivity or assessment, and it will search for you. There's also other types of skills or science skills that help support it. So you can see there are a bunch of pre-made things that also exist on the pathway. Things they've designed directly, so LabXchange, or things from Amgen scholars or other content sources, all exist in the library. Pause again.
So I'm just going to jump in real quick here, Mary. One of the things that several of us do is we will make a pathway of assets that for us, so they were sort of making libraries for ourselves. And so if we can go through and just pull out a bunch of assets, and that's one way we can just keep track of everything that's there for either protein chemistry or Beer's Law or something like that. So I know several of us do that as sort of a way to store things in an organized manner.
And we have a new question from our participants. Is there a limit to how many items can be in a pathway?
I think there is a technical limit. It's pretty high. I think it's like 30 to 35 or something like that. So if you're using it just as a folder, then yes, you might need to create multiple ones. But generally, I think of it more as like a lesson that I might give to my students. So, I try to limit it in the number of things I put in there. But I think the limit's around 35, if I remember correctly.
It seems like for a pathway for lesson, keeping it under 10 seems to be the optimum amount that I found. Otherwise they're just overwhelmed with the number of assets they're having to deal with.
That's a great point, Dave. And I have it's sort of along the same lines, and you showed a little bit of you showed a possible solution with the remote learning cluster that you showed. But have you heard any kind of common challenges, Dave, from your perspective? Are there common challenges you've run into when working with LabXchange, or that, Mary, that you've heard about from other teachers in working with LabXchange? And what advice do you have for teachers as to how to address those challenges?
I think the biggest thing that I struggled with, and that I've heard from others, is the search engine is difficult to get to focus in. So that's one that's gotten better. But it's just trying to narrow what you're looking for. And what I have found is I sort of do my big umbrella topic and then go in and look for the type of asset. So if I'm looking for a simulation or a scrolly, and then after that, if you know sort of the creator. So one of the powerful things about the LabXchange is it's just not things created in-house. They're starting to collect from all other groups. And so if you know a little bit, either it's FET or Concord Consortium, Amgen, that can also help narrow. There's just so much out there that having some strategies to sort of focus your search really helps.
Which is a great skill I think all of us are finding overall as the amount of information out there grows so exponentially. Mary, for you, any challenges that you hear?
Yeah.
Recently?
Well, we're a BYOT school. So because students are all bringing different kinds of devices, some of them have really small screen sizes. So, or trackpads that don't work, like everybody has something, right? So one thing I found helpful in the simulations is a nice gem that they've created for accessibility reasons, right? Like if you have a really small screen, let me see if I can, as you resize this, you can see this screen kind of changes what I can see. So one thing, if their trackpad isn't working or they're having issues like clicking, if you use the tab, you can kind of see there's like a pink box or a pink outline that's appearing here. You can tab through to select these different pieces. I'm just hitting tab and enter, so that if it's something that moves, like one of these micropipettes, it brings up this menu. And then the menu you can tab through to decide like, okay, I want to move this micropipette to the P- tube, and it'll automatically do some of the moving for them. So that's great for students who have like smaller screens or broken computers, or, for whatever reason need to use keyboard controls, or a screen reader can interact with this component as well. So that's one of those tricks. Like sometimes the physical like clicking through of like moving things around and doing the lab virtually can be a little tedious, but those are some shortcuts to help with that. The other thing I would say is the links, right? Like now that everything's digital, students have like a million logins and they have to go to a bunch of different sites. So to me, that shortcut of just sharing out the URL from my class that I've created sends them back to the same place every time. They don't have to like remember a million logins. it kind of funnels them there. So if you share the URL from your classes page, that will help sort of streamline that activity.
That is a great tip, Mary. And I wonder, I know we have one of our master teacher fellows, more than one, is actually interested in the development process at LabXchange. So can you, I don't know if you can speak to this, but can you share a little bit about what the, when LabXchange decides to develop something new, like a simulation? And Dave, you work with them as well. So what is the process kind of from idea to reality? What's the journey that happens to create an asset or a simulation?
It very much, oh, sorry, Dave, you can go ahead.
No, go ahead, Mary. I was just taking a big breath to think.
I was going to say, it very much follows the process you would do if you're creating something for your classroom, right? It'll start off with the scripting phase. And usually what they decide to script depends on sort of what ideas people bring to the table. Like, is there a need? Like, do we need a model of something that doesn't exist? And they start with that. Like, really, they do ask teachers, like, what do you need right now? Sort of what isn't out there? And they ask people to script it. You'll create a script, which might be like a text document or a graphic, like slide script with pictures to help designers understand, like, what are you trying to visualize? What's the general flow of something you're trying to create? Or if you're doing a lab sim, you might start with the protocol and kind of think about what context you build around that. So once it goes through scripting, there's a lot of rounds of feedback. So they'll usually get feedback from what they call the content team. So that has teachers, they call them subject matter experts, so other scientists in the field, university professors, and other university staff as well. So they get feedback from not just K-12 teachers, but also university, because they're trying to bridge that gap and sort of span the spectrum of students. So they want to make sure it's aimed at a variety of levels. Then after they get the feedback on the content itself, they'll send it off to what they call development. And then the pictures will get made by graphic artists. They'll build sort of the motion and design between like those interaction points you might think like, hey, this isn't challenging, but to like make a micropipette move that has all these dials that you can set the volume, it is challenging to make it technically responsive in a way that's scientifically accurate. So that's another piece. They like try really hard to make sure all the things model or match an in-person experience as much as possible. They want students to be able to change all the variables. In the one where you have pelleted cells, like they show the pellet being created after you centrifuge. So all those little details are things that you have to communicate to developers who often are non-scientists. And are learning science all the time, thanks to content creation. And then once they have like a model of this, so they'll mock up a working model of the simulation or an asset that has motion. And then we review it again. So the content team goes back through and is like, hey, does this match our vision? Do all these pieces, are they scientifically accurate? Are they meeting our learning goals? And then they'll push it out for quality assurance. The writing team will go through and make sure that it's grammatically correct. The accessibility team reviews it to make sure that it's accessible for both like screen readers, but like visual components. They've carefully chosen a color palette to make sure that everyone can access the colors. And then it'll get put out and made public. And it's constantly undergoing user testing as well to make sure that it's meeting the goals that they've sort of outlined. So it takes a little while. Simulations are the biggest.
Mary, thank you so much. That was the quickest explanation I've ever heard. It was wonderful. And I know we're at time, so we need to wrap up. So I want to start by saying a huge thank you to our panelists, Mary Lou and Dave Welty. And we hope for all of our participants, this gave you some ideas how to use LabXchange for your own classroom. You're welcome to visit us at the ABE website. And we've pasted the LabXchange address in a few times. If you're not familiar with it, we encourage you to explore it. Next month, on the 22nd of February, we will be hosting another roundtable on real-world science, and we'll be joined by two of our Master Teacher Fellows from ABE Australia, Aidan Quinn and Joanne Donaldson, to tell us a bit about strategies they have developed. I want to say, take just a moment to say thank you to our ABE teacher community, and particularly a huge thanks to the Amgen Foundation, because they support both LabXchange and the Amgen Biotech experience. So we're very grateful to Amgen Foundation for that. We thank you all for participating and have a great rest of your day. And thank you for joining this ABE Teacher Roundtable. Take care, everyone. Bye-bye.